Well, thank you for having me. Before I start, I will mention a few things. So I am from a museum, and technology for most of my colleagues is a new kind of hammer, smashing fossils. So it's been really eye-opening being here and hearing, talking about the latest in sequencing. And we do actually do quite a lot of sequencing, at least in my group. But again, what I actually do mainly is bird genomics. So when Eddie wrote to me saying, would I mind giving a talk, I was sort of thinking, how do I type bird genomics into this? And thinking, well, we do something called the oil bird, which is this bird that's famous for you kind of stick it on a stick and light it, and it's like a torch. And I thought maybe, engineering the oil bird or something. But then Eddie clarified and said, no, I want you to talk about your other hobby, which is basically how we monitor biodiversity using unusual substrates. Now, again, this is my preliminary title, and then yesterday, as I was sitting through talks, I then realized that, of course, you guys all do biodiversity, but it's just microbes. So I thought, well, okay, I'll call it eukaryote biodiversity. And then the fungus guys ruined it, so now I have to do big eukaryote biodiversity. So this is what I'm talking about. And again, I'm not an ecologist, unfortunately. I'm a biologist, a museum kind of guy. And the topic we're actually interested in is basically to do with things like this. This is a nice picture of mammals. I guess you all recognize that, even though they're not bacteria. There's about 5,500 species of mammal out there, and that's pretty cool. Everyone likes mammals. They get on Discovery Channel and things. And if you look at where the mammals live in the world, this is a map that was put together a few years ago in a review. And the main thing is that if you can see, let me get back a sec. All these are gray regions here, these shading bits. These are what are called the mammal hotspots. This is where you find, in general, many more mammals than elsewhere. Now, the problem with these from a sort of conservation biology angle is that where all these stars are a tropical forest. And this is actually a major, major problem for conservation biology because although mammals are fairly big and so on, when they live in tropical forests, it's actually pretty hard to actually assess them. And if you look at some facts that have been put up recently by IUCN, since the 50s, 76 species have gone extinct. That's actually quite a lot. 29 may be extinct. Notice that word may. 25% of accessible mammals, this is about 1,000, right, are threatened with extinction. Again, note the word in red there, accessible. And 40% of them are threatened by habitat loss. And I just want to focus on what accessible means. This is from the IUCN, the guys who sort of do nomenclature for bigger things than microbes. And they have a ranking system here for how they describe animals and plants and so on. And at the worst case, they're either extinct or they're extinct in the wild, which means they're alive in zoos or in the basement of the California Academy of Sciences, I guess. And then you've got these various threatened categories, and they're listed as critically endangered, endangered, vulnerable. And people like to look at an animal that's out there and put it in one of these categories. But the thing is, they've also got this category down here, data deficient. Now, what this means is this is a species where we just simply don't know what's going on. And this is actually a major chunk of the species out there, not just mammals, but pretty much all animals. And it is a big problem, because if you don't know what's going on and where things are, it's very, very hard to implement conservation programs and to monitor what's going on. And again, people wonder, well, why is it so hard? This is just a picture of some rainforest for anyone who hasn't been there. It's very hard to see anything. If you fly over Africa, fly over the savannas, you can look down and see lions and zebras. It's all very nice. If you're in rainforest, you look down, you see trees. You don't see very much. And when you're in the forest, you kind of see things like this. Nothing. I mean, I've been in Borneo rainforest and literally been stepped on by an elephant. It came out of nowhere. We didn't even hear it. Very, very hard to see these things. And so how are people trying to do it normally? The kind of standard technique for looking at these animals and assessing where they are and then forming the basis of your conservation plan is rangers. This is what Google interprets a ranger when you do the first search. He's a Russian ranger. And these guys are, of course, semi-trained. They could be more trained. Generally, if you're very well trained, you're taken by another organization, like the army. So these are the kind of army rejects in general. And these guys sort of wander around the forest and so on, and they, look at things and say, oh, I saw an elephant, and they give records, and that's great. And another conventional method is I guess, feces. That's a polite way of putting it. This is animal feces, and people go out there and look at it and go, by the fruity aroma of that one, it's a brown bear, and you can look at it and say how long ago it was there. Tracks is another classic. If you can see this, there's an animal track out there. Camera traps are a very, very growing thing in conservation biology. They're essentially these infrared cameras that get tripped by animals. There's several problems with these. Firstly, small animals don't trip them. Secondly, locals steal them and sell them. Imagine you're in Laos, where the average salary is $1.50 a day and there's a $1,000 camera trap there, not many people will leave it. So they are okay, but they're very, very expensive and hard to include. Another classic way to discover animals is actually meat markets. Several of the recently discovered species have been found sort of chopped up in bits. Or as often they're found as like, antlers on walls. A guy, like a conservation guy, will walk into a headman's hut and go, oh, that's an interesting looking skull you've got there, and discover a new species, as happened with something called the Sowler recently. And so these are the standard methods, and they're not very, very effective for many, many reasons. So what we're trying to do is actually use metagenomic approaches to aid this. And I'm just going to go briefly into our recent success. This is a guy called Mads Bertelsen. Mads is a staff vet at Copenhagen Zoo. He is, without doubt, the smartest guy I know. He's kind of wasted in a zoo. He's also great. I mean, I guess it's not really relevant for you guys, but he's a really good dealer of animal parts. He's got no qualms. Animals die, and he'll ship parts of them everywhere around the world, which is very, very hard to get hold of. He really likes his research. Now, Mads had this idea not very long ago, and what he was doing was he was combining two ideas. He was in Southeast Asia, where they've got a research site, the zoo, and they're monitoring Southeast Asian tapirs. And while he was down there, he basically found a leech. I can't remember if it was on the tapir or it was on Mads, but in one of the two cases, he found a leech, and he suddenly thought, leech plus tapir, I wonder what the leech has got inside it, I wonder if it's got DNA, and I wonder if I can become very, very famous. And here's a gratuitous picture of another leech. These are the kind of things that you often get in Southeast Asia. They're big, big animals, but they do range from very, very small to very, very big. And leeches are a pretty neat animal when it gets down to these kind of things, because what they're sort of geared to do is they're geared to, first of all, seek out living things with warm blood, especially if it's things like male crotches, armpits. This picture here, I actually found it on Google, and I wrote to the person who took it and said, oh, that was a crazy big leech. And she was, yeah, my boyfriend found it on his arm or something. And I was like, oh, that must be horrific. And she said, well, it wasn't as bad as the one on his eyeball, which gives you some kind of like... And then I actually told the guy in Madagascar this, and he was like, eyeball, nothing. I had a leech on my eyeball. So they're... And I've even got a PhD student down in Australia right now, and she's actually going out hunting for these things, and she confirmed they really do get everywhere. So nasty, nasty animals. They're like homing missiles for mammals, but they do bite you and drink blood, and they drink a lot of blood. And then, of course, they sit there and they reproduce and they store the blood. And so Mads thought, well, maybe if we collect these leeches, which in theory isn't very hard, you go out there and stand naked for a few minutes and they jump all over you. And it's actually true. One of our big problems with leeches is catching them before they catch you. Otherwise, you end up sampling human everywhere. So anyway, there's a leech. So, I mean, he came to me and I was talking. I like to do control experiments. So I said, well, before we do the expensive trip to Vietnam, why don't we do a zoo experiment? So we got a goat. It's got an interesting Danish name like Meyer or something, this goat, from the zoo. And we bled the goat, and we then bought a load of medical leeches. So these are your good old Hirudo. They're a very different kind of leech. You can buy them in catalogs. They're, used to relieve your vapors if you're in a 17th century novel, that kind of thing. What we thought is, well, here's an easy experiment. Let's take the leech. Let's take the goat blood. Let's feed the leech, because we know these things can't stop pulling blood out of us. And then let's look at things like how long does the blood survive for, how much DNA can we get out, and so on. All very, very simple in theory. So what we did is we first of all got the leeches, and we thought, well, how do we feed these things? And we did a bit of Google searching, and it turns out people are trying to feed leeches in the virology field. And what they do, it's brilliant. They apparently get a condom, and you fill it with blood, and then you kind of put the leech on the condom, and then it drinks the blood, and everyone's happy. So we tried that, and it's kind of like having a water balloon and biting it. So except that you have full of blood, so the leech comes on, bites it, bam, you've got blood all over you. Not the most effective method. And it actually turns out leeches are nearly impossible to feed, despite the fact that you can't get them off you in the field. We spent a very long time coming up with a this is our idea of high tech, right? So in the last talk, you guys saw cool microfluidics. We take falcon tubes and cut them in half and put a bit of parafilm on them. And so we have a parafilm covered tube, we have a syringe, and we basically use the syringe to force the leech against the blood until it can't get out and has to eat its way out. That's how we feed leeches. So we did this, and then we let our leeches sit in the fridge for a certain amount of time, and then we killed them occasionally, and we did a real-time PCR and various things to see what's inside the leeches. Is it goat DNA? We hope so. And how long is it there for? And we can make a nice decay curve. And the important thing was we started with about 40 leeches, and we kept them for up to 4 months, thinking that's plenty of time. After 4 months, they were still full of DNA. I mean, it doesn't look like it on here. This is time, of course, and it looks like there's nothing left. But if you look at the amount of blood and DNA in them to start with, they've still got like thousands of copies of DNA per microliter. So that was very, very promising. And what's more, we could actually amplify like 5 kb fragments of mitochondrial DNA from the goat, from the leech after 4 months. So that was really quite promising. We thought, well, that's great. If a leech has eaten somebody, that's out in the field, and we get it, maybe we can get a lot of information from it. So at this point, we went out to Vietnam, central Vietnam. We collected terrestrial leeches. And we did a very, very basic experiment. We didn't use any of this next-gen rubbish at this point. We simply did PCR and cloning, and we wanted to see what's in them. And we had about 25 leeches in the first batch, and we were thinking, if we're lucky, one of them will work. And it turns out that I think about 22 of them out of 25 gave us mammal DNA. This is just with generic mammal primers. And of these different things we got out, we actually got some strange things. This is the Annamite striped rabbit, discovered in the last 10 or so years. It is data deficient. Nobody knows anything about it. It's thought to be very, very rare. It was in 5 of our 25 leeches. It's not meant to be in the region. This is a muntjac, exactly the same story, discovered 10 years ago, meant to be very, very rare in about 3 of our leeches. This is a mainland siro. It's a kind of deer-like animal. Ferret-toothed badger, a very bad-tempered animal. There's 3 species with different length teeth. You can only measure which one it is normally by grabbing it and measuring its teeth while it's trying to eat you. Not very, very easy. But we found it, and that was pretty cool, and we were very, very lucky. These things have never been observed on camera traps, and they've been trapping for about 1,000 nights, very, very heavily. So the point was we were suddenly finding all these animals that are meant to be not there, meant to be very, very rare. And this, of course, got us very, very excited. It's a cryptic species. And I'm going to stop on leeches for a second and just go to some other substrates, because the idea is we can do it with leeches. What else can we do this with? And the general idea is if there is DNA in something, after a certain amount of time, the DNA may still be there. Can you get it? And these are the kind of classic substrates people are looking at to try and see what's going on. Anything to do with the diet, like feces, for example, we'll often have it in, so defecation. Urination, when an animal pees on the ground, it will pee out DNA, and you might be able to find the DNA if you analyze the ground. A lot of shedding hair, shedding skin, and so on, and decay. And this whole idea of using environmental DNA for a non-microbial context was really kicked off by this guy here, I think. This is Esker Villerslev. He's this guy who runs the center I belong to. And what Esker and his colleague Anders did in their masters, actually, and then their PhD, was they went to Siberia. They're field guys. They like being in Siberia. And they took soil cores in Siberia. This is a permafrost, so the idea is, well, it's frozen. Frozen means like a freezer, so if there's DNA in the ground, it might survive a long time. They originally tried coring with sort of vertical cores. Turns out that's not very, very easy, so instead they worked out a much simpler solution. You make a very, very small core on the end of a very, very big drill, and you drill horizontal into these big permafrost sheets on cliffs. You get a small sample like this, and you extract DNA from it. And of course, you can look at bacteria if you want to do it. And in fact, they have done that. But he was interested in, can we find environmental DNA from things like the plants that were growing there or the animals? And he published this first paper. And all that was important in this context is that they have samples which they could age using different methods like carbon dating and OSL dating. These different colors represent different kinds of plants that they were amplifying using plant barcoding primers, and through time they could see the plant community changing. So this actually caused quite a bit of noise, because the idea was you can actually track back through time what's in the environment by DNA just shed by the plants, whether it's like little fragments of roots or so on. So that was pretty cool, but that was in Siberia, which is difficult for 2 reasons. Firstly, because it's in Siberia, and you have to bribe a lot of people to do anything. And secondly, because it's in permafrost, which is great, but only over a certain part of the world. And so we wanted to know what happens if we actually go into non-permafrost regions. So we ran a load of experiments, and we took advantage of basically zoological safari parks. So if you want to go looking for environmental DNA and you want to tie it to questions, for example, how is it put there, or is there a relationship between biodiversity and what's there? It's very hard to do in an area where it's very hard to control the animals there. But the nice thing about safari parks is they're full of animals which are not native to the country. So if you find DNA from the animal or the plant, like in a botanic garden, you can actually say, well, we know where it came from, and you can do various things with it. So we had a little project. We went to some zoos, we drilled some cores, and we found some interesting results. Firstly, these are various animals that don't live in Denmark, at least not for the last 50,000 years or so. Ostriches, various kinds of cats, elephant, and this is just a soil profile. And by taking sections of the profile and trying to amplify DNA, we could find that actually the DNA, if you're an elephant, goes down a very long way. Does anybody ever hear like YouTubed elephant peeing? You'll know why. They release something like 20 liters of pee at a time. That just goes straight down into the ground. but it does decrease in frequency. Ostriches, on the other hand, the DNA doesn't go down very fast, but if you look at what bird poo looks like, you'll understand why there's not much liquid to it. The cats are interesting because in some regions there was no DNA, and in other regions it went down a long way, and we were like, well, that's kind of weird, until we Googled cat biology, and tigers, for example, have these latrines. They like to pee in very, very specific spots, and in fact, where they do that, DNA goes down. So that was interesting. We could see that, the DNA is getting there from the defecation and so on, and we could also do a quick assay on biomass. We know the biomass of these sites because we know how many impala live in the site and so on, and we know how they've been through time, and what you can then do is simply look at the biomass of the animals, the number of the animals, and the amount of DNA in the soil, and correlate them. And there is some kind of correlation with some species, in particular between biomass and the amount of DNA. So that's kind of cool. It suggests it's a fairly reliable method. We then tried this in various other environments. This is in New Zealand, and in New Zealand, there used to be something called the moa. Moas are like very large ostriches, but they got eaten by the Maoris very, very quickly. And by taking cores in New Zealand, we found interesting things. This is going down through time. You find different moa species, big ones, little ones, intermediate ones. By coupling this with the plant community, you can see when there are big moas, there is short grassland kind of environment. When there is little moas, there's more tree-like environment. So you get these nice correlations. Being New Zealand, you also find sheep DNA everywhere because sheep just pee on everything in New Zealand. But so that was quite promising. Another thing we tried to do was say, well, how useful is this really? And one of the big questions in sort of Pleistocene biology is, why did there used to be a load of mammals and why are they all gone now? And things like the mammoth, of course, used to live everywhere, and the fossil record says they died out about 10,000 years ago. Now, at this point, Rasmus Nielsen stepped in. I guess a lot of you know Rasmus. He's a bit of a math statistics kind of guru. And Rasmus did a little calculation based upon the fossil occurrence and what that means about population density, with the idea that the fossils you find do not represent the last animal. There's a sort of a probability. The fewer animals there are, the fewer fossils there are, and so on. And although the fossils stopped 10,000 years ago, the animals may persist. And Rasmus sort of did some kind of calculation suggesting just because the fossils stopped 10,000 years ago doesn't mean that the mammoths weren't there. So what we again did was we took cores in Siberia. Again, this is time. These are carbon ages, 7,000 years onwards. The important thing is, at about 7,000 years ago, in this case Alaska, sorry, we're still finding mammals in the soil. Now, this course could be DNA, but we tend to think the DNA moves downward with the P, not upwards with the P. And a lot of controls are run, but it does seem that the DNA in the ground is suggesting they were there 3,000 years longer than the bones. And this has lots of implications for guys who care about mammal extinction. So that's the kind of stuff we do. I just wanted to show you this nice picture. This is reconstructing ecosystems. This is what we'd really like to do it with dinosaurs. The only reason I show this is because, of course, there is a limit. DNA lasts for a certain amount of time. Depending on the temperature, it may be anywhere up to 1 million years, but we're certainly never going to get back to dinosaurs. The biggest kind of questions we can do time-wise are things like Greenland. This is Greenland today. It's very, very cold, not a very pleasant place to live, quite beautiful. By coring through Greenland and looking at the plants and so on, you can make pictures which nearly rival Joe DeRisi's picture of the snake yesterday. This is the one the journal produced. There's butterflies and everything. Unfortunately, no dying, bleeding mice and so on. But you can start to make these estimates of how the environment changed. This is with sediments, but there are other sources as well. Another classic one we've worked on recently is freshwater. It turns out if you go to freshwater ponds, you can collect DNA. And you can look at the DNA from the water, and this is, again, DNA from eukaryotes, and you can actually see what kind of things are there just by generic barcoding sort of things. And in this case here, we looked at DNA from various amphibians and fish and invertebrates and so on. And you find that, for example, 100% of the ponds where we know there are frogs have got DNA from frogs, and 90% where we know there are these lacewings, there are lacewing DNA. And then these light gray ones are ponds where we think there might or might not be animals, and it turns out the DNA is often there. You can also find out by we did experiments where we basically made fake ponds that actually the level of DNA in the water correlates very nicely with the biomass of the animal there. So this is another way we can go out there and survey what's in the water, and this method is actually working really remarkably well. This also works on seawater. We actually did some test experiments in pools where they have trapped porpoises, and we found they were full of porpoise DNA. And as we went into the sea away from the pods, we found porpoise DNA surviving. And at one point, we actually took some water and we found DNA from something called a melon-headed whale. And this was kind of weird because the Baltic never has melon-headed whales in it. And we thought, well, there goes our idea of this working. But then it turns out a melon-headed whale had been through there a week ago, a very, very strange, rare one. That's what had been reported. So even seawater seems to work.
Another thing we like to look at is insect diversity. We do this in several ways. This is actually Doug Yu, who's at Kunming and UEA. What Doug does, he does these great experiments where you either fog trees and collect all the insects out of them, or he puts up winkler traps or the equivalent, and he blends up the insects, and then he sequences the blend of insects and just actually looks at what's in there through metagenomic sequencing of it. We actually do it using a slightly more targeted approach. We use bats, so we let the bats fly and catch the insects, and then we actually grind up the bat shit and look at what's going on. And this works quite well. Vampires work as well. Vampires are also easier to feed in your control experiments. Vampires do actually give you quite a nice idea of what mammals are in the area, and birds. So these are the kind of substrates. Of course, how we do it is the same way you guys will be doing fungus or bacteria, just simply tagging of the primers, amplifying them on the Flex, on the MySec, on the Iron Torrent PGM. We don't use that, but a lot of my colleagues do. A lot of my colleagues are poor. They don't have a lot of money. And the PGM appears to be the poor man's MySec, as far as I can tell. And it does work very, very nicely for this kind of amplicon work. Of course, all you need is suitable read lengths. You need to be able to discriminate what's going on. One of our big problems we have is that our DNA is degraded. And because it's degraded, we have to use very, very short barcoding regions. And this leads us into a lot of bioinformatic headaches. The kind of problems you will have with identifying what kind of bacteria you have on 16S data is compounded often with us because we've got 150 base pairs of something. So we're really, really fighting that. The actual process we use is the same as everybody else. We sort them, we merge, we resort. We often have to look for inconsistency between replicates. We do a lot of replicates to rule out things like sequencing error, especially because of the uncertainty of identifying things. The real challenge we're at right now is identifying what we find. Although there's only 5,500 mammals out there, no more than about 1,000 have even got these 16S sequences. So we often get sequences and we think, well, it's a kind of rat. And that's not a very good answer for conservation. So what we're trying to develop now is a sort of a probabilistic-based method where you sort of have Bayesian-like priors, and you can sort of say, well, I know I'm in Vietnam, I know this data's in GenBank, I've got this sequence out, it blasts as this, but with these priors, it's probably one of these few things. It's not an Antarctic kind of thing, it's probably a Vietnamese thing. And we do this because otherwise we spend a lot of time looking at Megan and look at colored dots and trying to make sense of that, which anyone who's tried that realizes is not the easiest thing in the world. Alternatively, we have to look at various bar charts and compare things. So we are, actually, we've got to the stage where we can get DNA fine. It's the interpretation of the DNA that's the problem. Another big problem we have is contamination prevention. These samples have got very little DNA in them. They're very easy to screw up, and this happens a lot, especially by enthusiastic new labs who want to start to do the work. In conservation stuff, you've really got to be sure you're right, because if you identify, for example, the Sowler lives in a certain part, conservation organizations will chuck all their money at it, and their money might only be $100,000, but then they can't, preserve penguins in southern Antarctica or something. So they really have to be careful with their money, and they are very, very prone to run off and do things on the spur of the moment. But of course, if you've screwed up and you've contaminated something, they're wasting a lot of money. So we spend a lot of time doing this. The other thing we're always doing, of course, is trying to bring down the cost, because we often are interested in sequencing DNA from thousands and thousands of these things, and it is expensive, and of course, we're at limited budgets. So now that we've got the methods working, we're actually at the stage of just integrating it into the monitoring. We can find out that animals are there, but what do we do next? We can find out plants to do there. And this is actually another big limitation, because there are many, many conservation methods out there, as I said earlier, the camera traps, the visualization, the going and asking people in the villages. So what's really needing to be done now is actually control experiments where many, many methods, the DNA, the non-DNA, the drones, the so on, are all put into one site and compared, so you can then say, for this kind of region, you want to use DNA and camera traps. For that kind of region, you want to use people on the ground and bribery, whatever you want to do. Bribery works really well, actually, incidentally. Back to the leeches before I finish. So originally we had mitochondrial data, and it was useful because we could say there was an animal. But of course we want to know more than it's just a Sowler there or it's just a ferret badger there. We want to know how many are there and so on. So of course the big question is, can we get larger amounts of data than the 150 bases we're used to? It turns out you can. You can actually very easily get mitogenomes of the prey from the leeches after 4 months, which is pretty cool. You can get microsat data from them. You can probably get reduced representation from them. Leeches have got the nice benefit of only eating really one prey at a time, unless they're disturbed. If it's a human they're trying to eat, they're often disturbed because a human has a panic and pulls it off their eyeball. But if it's a ferret badger, it ignores it. The leech gets full of... So it's essentially a very good pure DNA source. So you can actually get very, very good DNA out of it. And what we're really working on now is working ways to get through large amounts of these things. And the way we do it is actually we make leech soup. We get a load of leeches in a tube and we sort of blend it. And then once we've got the blend, we actually can analyze it. And then by doing it in the right kind of way, you can actually go back to the individual leeches and you can actually work out where things came from. And that's actually working out quite well. So I'm going to stop there. Just a quick thanks to Eddie for inviting me and, of course, Marsha for doing all my logistics. These are the main guys behind this work. Ida Schnell was actually a master student who did all the leech work, and Andy and Philip are the soil and water guys, and these are the back guys. It is actually a method which we're hoping will take off, and we're hoping in the near future we'll actually be able to use it and get useful information. From the micro point of view, I would suggest it's worth thinking about because the DNA is being preserved inside these leeches in the crop. There's something funny going on in the crop. I mean, in no other situation in a 30-degree temperature does DNA survive with 10 kb fragments for 4 months. So there's something funny going on in that part of the body which the metagenomics guys might want to look at. So with that, thank you. I'll stop there.
So I had a few questions. So these leeches then fall off their prey, and then when you go through, they're looking for a new prey?
They are looking for their next dinner, yeah.
And I'm thinking about things, so if people aren't from California, there are a certain group of people that think Bigfoot's run around in the woods. And so I'm thinking about ticks as another...
Yeah, ticks work, mosquitoes work, carrion flies work, but the only problem with them is they have a much lower success rate because the DNA doesn't survive so long in them. On the other hand, if you do get the DNA, you know it's very, very recent.
Other questions? Behind you. Behind you.
How does the leeches compare to cameras?
How does the what, sorry?
How does the leeches compare to cameras? You know, will you see things, you know, you have data now, and so did they see the same things using these
The leeches tend to see more, actually. So a lot of these animals that we're finding, the cameras don't pick up at all. So they did 3 years of camera trapping at the site, never saw these rabbits, and then the leeches had them.
And do leeches have a host range?
These are the big unknowns. Leech biology is unfortunately very poorly understood. You don't get big grants to do it. And the world's leech expert is at the American Museum in New York. He laments this fact, but we know almost nothing about them. When we find leeches, most of the ones we find, he goes, oh my God, that's a new leech. So there are a lot of unknowns. Big dietary preferences could be a big problem. It could be through age, for example. The young ones might eat different things to the older ones. So you do have to watch out for this kind of thing.
And how many leeches did you survey to get to these rare, nearly extinct organisms?
We did, I think, about 20. So it was actually the first 20 we looked at pulled up these 2 unknown organisms and so on. They also pulled up a cat, which we brushed under the carpet because we were spineless. But it was a cat sequence which didn't match any catch in GenBank and may well be a new cat or an unsequenced cat.